alright. So for me macam I really want to assist and help, alright. So but the thing is you guys kena tolong diri sendiri dulu, then I can assist you. You tolong yourself untuk try to answer some questions, and then I can try to give you some marks according to your efforts lah, alright. So I'm not saya tak marah, saya tak apa, cuma I just want to give some advice lah if possible. Sebab some lectures might be more strict, right? Even soalan salah pun apa semua dia akan straight blank lah, right? For me, I will try to find and seek some answers for you guys, but this is me giving some advice for you guys, okay? Cuma saya tak boleh bantu for the final later sebab I'm not in the final, alright? But I can try to assist you as much for during the mid semester lah, alright? So this is where it will leads to our punya main introduction for today lah, right? Can I share the screen ya? Mas, remind saya untuk apa nama? Untuk record every session lah ya. Sebab I find that kalau saya record it will be more beneficial for you guys. Korang boleh trace balik lah. Alright, so allow me to record the session with everyone ya. Mana record button? Dah tak ada record Tak ada record mana record button? Screen. Hello, dengar saya ke? Let me share sikit.
Dengar.
Alright. Tolong minta maaf. I want to record the session but macam tak jumpa. Nampak ke?
Nampak.
Alright, it's good. So this is the thing that I want to guide you guys a little bit lah. Alright, dikit saja before we start our main session. Alright, so the main session will be start within around 3 to 5 minutes later lah. Kita nak bincang pasal course outline a little bit for you guys, and what are things that you guys, strategy that you guys can do for apa nama for to try to get some marks for final lah. I mean for this particular course. Right? Saya tak jumpa lah untuk record, but it's okay. Let's just start here first lah. Alright. So this is the subject. It's second year subject. Right? So it's SAP 222-3. Right? So this will be your course learning outcomes. Alright. So explain the basic principle of Mendelians and molecular functional genetics in plant. Illustrate the historical aspect and role of plants genetics in relation to crop improvement. Present the principle, knowledge, and tools related with plant genetics. Right. Let me tell you one thing. Most of the questions and most of the assessment, most of the lecture will be guided by the course outcome, okay, course learning outcome. So you guys can try to predict some of the questions and some of the answer from here, right? From this particular topics, right? So these are the topics that will be out from this particular session lah, right? So this is other topics. So maknanya you guys can try to predict, right? Certain questions ataupun certain assessment from these particular topics ataupun related with this particular topics lah, right? Mendelian, molecular functional and genetics, historical aspect, for example, right? And then present and then principal knowledge and tools related, right? So kat tepi ni tak perlu, jangan risau. No need to understand about this part yet. Try to check here. So not just my subject, but other subject as well, right? So for example, if you guys realize Macam plant physio there's one course learning outcome ni related with phloem. That's why ada satu soalan related dengan phloem. If you guys realize lah. Right? So that is the course learning outcome from there. Right? Try to, bukan predict, try to focus more on this particular area. This is what I mean lah. Right? So the next one, this is our teaching ataupun the assessment lah. Will be lecture, laboratory practicals. Nampak tak ni? LOI 1, learning outcome 1 ni. Alright, this is the one. So we will have the practical session dengan lectures. 3 pun sama practical session dengan lectures. So which mean that dekat sini, we will not be there. Dekat practical session. And you guys can realize dekat sini kita ada assignment, ada presentation, lab report, mid semester exam dengan final exam. So this is what will be some of the outcome for the midterm and for the exam. So which mean that LOE 3 ni dia dekat final saja. Alright, which mean that this one present the principal knowledge and tools related with plant genetics. Alright, so you can try to strategize your planning lah. Strategize your planning on how you can outcome this particular session. So that's why in early of the week lah, biasanya kita akan provide you guys with the course outline lah. So that's why you guys can read it through, bukan read lah, ataupun analyze lah. Tengok what is the strategies for. Alright, so this is the most important part dekat sini lah. So mid semester exam will be 20 marks, right? This 20 marks is pure 20 marks for you guys punya total course punya marks later. Same goes with final exams, right? 40%. So which means that out of 100% from your final marks, right? Out of 100% from your final marks, right? It will be 40% from that. For example, if you score 100%, right? 40 marks will be sum up to your CAM. Right? CAM is normally around 60 lah. 60 including 20% midterm, 25% laboratory, 10% assignment and 5% your presentation. So this is where you guys can have the mark allocated for the course. Right? So this is total up 100% for the course punya marks. So this is where you guys can see the cumulative punya marks lah. Right? So dekat sini, saya nak highlight dekat awak semua lah, korang semua. Perasan tadi, this is 25% for laboratory. Right? So it's very very huge kat sini sebenarnya. Alright, that's why I tell you guys, I want to try to assist you in how to answer some of the format dengan questions dekat dalam laboratory. Alright? So 25 markah ni sangat besar tau. Right, we got 4 lab session, right? If you submit all, you are guaranteed to have marks. Right, kalau hilang satu memang awak akan hilang sebab you will be average about 4 lab session. Right, so 20 marks ni lebih besar daripada midterm. Right, so that's why I want you guys to try to focus at least for the lab, assignment and some presentation. Kita tak ada quiz ya. Right, so presentation ni try as much as you can do. I saw you guys the things that you guys did last semester, so I think that is good. So you guys can do much better this semester, right? So we will have some presentation and some assignment as well. So I will give you some topic for assignment and presentation later early on, so you guys can have more preparation for that. But for the laboratory ni kita ada 4 kita ada 4 laboratory. So this is me and Dr. Zarina discuss. So some genetics matters, you need to do the hands-on things, right? So that's why the marks is quite huge dekat sini. So around total around 60 marks from here. 5, 10, 25 and 20 will be 60. Alright. So this is where you guys can try to strategize how to score for your course. Bukan final for your course. Final just 40% sahaja. But it sum up a lot sum of your marks for this particular subject. Alright. So I hope with this particular explanation, right? So you guys can have some ideas for your semester on this particular, for your course for this particular semester. I mean, but we'll try to tengok balik. If you guys realize lah, dekat course outline plant physiology tu memang ada salah satu dia, if I'm not mistaken, CLO2. Dua-dua dia memang related with phloem. So that's why if you can try to focus on phloem last time, maybe you can try to score on that particular questions. Something like that lah. Right? So any questions regarding this particular course outline before we start our session? Any questions?
No.
No.
No terus. Alright. So can we start with our class session? Is it okay?
Yes, Doktor.
Alright. So yes saya
Tak boleh isi attendance.
dah limit
Tak lah, dia memang tak ada tadi. Dia tadi.
Dia kosong.
Serious? Oh, okay. Kejap. Let me try to edit it first lah. Sekejap I know what happened. Because I need to put some record for that. Bear with me for 1 minute Sekejap. Everyone can log into iTaklim? Boleh masuk ke?
Boleh.
Sebab if I'm not mistaken, yesterday ada sedikit issue for regarding internet dengan iTaklim, if not mistaken. Alamak. Tak boleh masuk pula This is what I mean. Dia still loading dan tak boleh masuk. Taklim. It's okay. Maybe after the class session, I will try to reopen it and I will inform Mas for you guys untuk log in. Is it okay? Boleh ke?
Boleh, Doktor.
Alright, thank you so much Mas. Sebab still loading tak boleh masuk sebenarnya. Alright, dah boleh kok. Let me see. Give me 1 minute Mas, kita try untuk habis dalam masa pukul 10 Lagi 40 minit. Is it okay?
Okay, Doktor.
Alright. Memang ada problem lah. It's okay lah. We proceed with the class first lah. Alright. So then we go for that. Boleh nampak ke my slide?
Boleh.
Alright. Let me try to make it full page. Full page. Okay. Ada je. Dah. This one okay. But it's okay. Alright. So let's stop. Ni boleh nampak ke semua? Semua boleh nampak Alright. So this is the SPL 2243, our previous subject, our previous program. And now we have the SAP 2223, right? The applied plant science, right? So this is regarding plant genetics. And we are in week 1, we will talk about a little bit of history of plant genetics. Okay, if you guys realize, history of plant genetics ni dia ada dalam CLO 1 dan a little bit of CLO 2, alright? So try to focus on these particular areas. It will be good for you guys, right? So what actually is plant genetics? Right. Plant genetics is a living organism. Plant is a living organism typically grown in a permanent site. As I always did mention to everyone, plant ni tak macam other organism lah, right? Other organisms, they are mobile, right? They can move from one place to another place. For example, if they are under certain stress, for example, right? Contohnya dalam keadaan panas ke, ataupun flood, banjir, ataupun inconvenient environment, alright? So they can try to move from one side to another side. Contohnya panas, dia akan cari to shades areas. Kalau banjir, dia akan try to cari tempat yang less watery areas and so on. However, plant is different, right? They will always stay in one permanent site and they can move ataupun mobile by spreading their punya spores ataupun spreading their punya seeds from elsewhere, far from them, right? Through a lot of means lah. It can be biotic or abiotic punya means lah, right? Biotic ni macam monyet, bees and so on. Abiotic ni maybe from the environment themselves, right? Angin and so on, right? They absorb water and some of the inorganic substance like the metals, right? Through its roots and synthesizing nutrient in its leaf by photosynthesis using chlorophyll. But I guess you guys learn all about these particular terms in the plant physiology and EPS lah, principle of plant science last time. Right? So this is the thing. And genetics is the branch of biology that deals with heredity. Right? That deals with heredity. So, oh nak record Nak try record. Oh lupa. Thank you. Let me check back. Can I stop the presenting and I will try to start the record first, yeah? Kenapa tak ada record button? Dah tak ada record button ke apa ni? Sebentar. Alright. In the activity areas. Okay. Yes. Sorry, sebentar. Can we try to make the record session? Kenapa tak ada record button ni? Hold on. I know, right? Let me try other means. Sebentar saya try record guna VLC lah. Awak ada button record ke?
Rasanya dah tak ada doktor.
Tak ada.
Dah tak ada kan? I think
Ni apa
any features daripada ni ke dia tak bagi orang record. Dapat record? Tak ada kan?
Dia dah tamat 10 Januari.
10 Januari dah tamat It's okay. I have another ways untuk buat sekejap Tapi bila saya buat pakai VLC ni, dia file jadi besar tau. Sebentar Alright, I know. Make media. Capture device. Desktop. MS. Sebentar I will try to make the record for you guys. Alright, dapat dengar ke? Hello?
Ya, doktor.
Alright, so now I record the screen. So now let's have the session now. Alright. Okay, nampak ke? Boleh nampak?
Boleh, doktor.
Alright. So bear in mind this is a screen record. Whatever happened in the screen, I will just record it for you guys. So I try to give it to you later lah. Tapi this particular file is a little bit huge, besar, so maybe around 2 or 3 gigs. But let me try to reduce the size for you guys lah later. Alright. Let me talk with Mas later lah regarding that. Alright. So this is you guys can see dekat figures ni lah. Saya sambung ya. Alright, so this is Solanum lypocusum, right? Lypocicum, right? So Solanum lypocicum ni adalah tomatoes lah ya. So this is micro tomatoes, right? This is the size of the falcon kita punya PCR tubes, not PCR tube, 1.5 ml tubes, right? So this is the size of the matured tomatoes, right? So to be honest, this was taken in my laboratory last time. During my master and PhD studies. Memang dia besar ni saja dan dia keluar buah besar ni. Right? Dan kecil is quite nice to put on the desk lah sebenarnya. So my main study last time we are using this micro tomatoes. So the reason why kita do some research regarding not getting the genetics lah. But the reason why kita menggunakan this particular micro tomato is because of dia punya short maturity period. Alright? Dalam masa 8 minggu dia dah boleh start to use ataupun kita boleh buat eksperimen for them lah. Alright, kita boleh dapat bunga dia, dapat matured plant and kita dah dapat dia punya hasil lah. So my study last time is regarding parasite lah. So kita infect dia punya akar and see what is the actually happen dekat dia punya tumbuh-besaran pokok. And what are changes inside them, what are genes that been upregulated and what are genes that we are targeting for. Right, so this is the actual pictures for getting micro tomato. So why I put here in genetics is because of it deals with heredity. So this is the strain ataupun this is the traits that have this micro size of tomatoes. Alright, so compared to other tomatoes as well. So this is different varieties lah for this particular tomatoes. Right, so heredity is the passing traits from parents to the offspring. Right, so maknanya whatever what the parents have, right? They will be inherited by their offspring. But we will study more on how this particular genes ataupun this particular traits will be inherited to their punya offspring later, right? So traits are the characteristic of an organism, right? So such as a domestic cat, for example, dia punya size, dia punya fur color, and dia punya fur pattern as well. I believe maybe not more, but some of you guys have pets, for cat pets as well. So you know the offspring lah. Kita tengok ayah dia daripada mana, mak daripada mana, and then you can see how is the breed goes. And sometimes, right, we cannot find any trait that coming from the mother or the ataupun father lah, but it appears in dia punya anak dia. So that most probably is coming from the previous ancestor ataupun dia punya grandfathers ataupun grandparents lah yang akan dapat. So there are some features that can be skipped from the generation to the next generation, right? So this is among things is quite unique lah sebenarnya, you can say, right? So plant genetics is study of heredity in plants, specifically on the mechanism of heredity, transmission, variation of inheritance characteristics. So this is what we will study in this particular course sebenarnya in general lah, right? So the next one, I think you guys know about this. Why study plant genetics, right? So provide basic principles for Mendelian, right? Transmission and molecular functional genetics, right? So this is the CLO just now, right? Provide historical aspect and role of plant genetics in relation to crop improvement. All right. So I believe you guys know in this particular world, I think you guys have you guys ever heard about golden rice? Pernah dengar about golden rice tak? Hello?
Golden rice.
Nah, yes. So the golden rice is where some scientists try to improve certain traits ataupun certain uh, aspect in this particular plant. For example, this goes this way. In the African countries, certain region ataupun the third world countries, some of them are having lack of nutrient ataupun we can say the malnutrients punya areas lah. Alright, so what happened adalah they found out that in this particular region, alright, they are lacking of vitamin A, right? So in order to get the vitamin A, they need to get it from a different type of sources lah. But due to the condition ataupun the environment, there is not really convenience for them to try to get some of the food source of foods that can have that particular nutrients. So scientists try to solve these particular issues, right? So basically what we do and what scientists ataupun anyone else, they want every time they do the research, they will try to assist and solve certain problems. So the problem here is that the malnutrition among the people that live in the particular areas. So what happened adalah they improve the values of rice dekat situ. Why rice? Because that is makanan ruji of that particular people's ataupun communities there. So senang, 2 in 1 lah. So while they are eating their punya makanan ruji, right? So they can have 2 things at one lah. So they can have that they punya particular foods yang dia selalu makan and with the improved values of that particular foods. So this is where the golden rice coming lah. So this is what we need to know and we can try to understand. We can try to increase certain values for that, right? So this is where we can say that crop improvement as well. Not just genetic modified, yeah. So we can try to breed them, crossbreed them to get some of the input, right? To understand the principle, knowledge, and tools related with plant genetics, right? So as you guys can see from here, right? This is the type of maize, corn lah. There are different type of values, nutrients as well, right? So dia ada berbeza-beza. So the taste of course will be a little bit different. Right, I think I believe you guys pernah makan jagung susu. Pernah makan jagung susu? Pernah?
Benar.
Yes. So in certain area jagung susu ni I think I was when I doing my study as well. Ada satu jagung tu memang dia colour putih. I think it's like here lah, jagung susu. Even that particular jagung at that particular place that my study last time, you can eat it raw. Makan dengan mentah je pun, you can feel it's already good enough, right? Dan kalau masak lagi lah sedap lah, right? So this is quite interesting, right? To get that particular traits lah, right? And increase also the value of the products, right? So I think jagung susu is far much the price is a little bit pricier compared to dekat I mean normal type of jagung lah. Alright. Sorry. So kita sudah 2 kali pula. Sorry. Alright. So next we go to the history of plant genetic. Alright. So pernah dengar nama Gregory Mendel? Have you guys heard about Gregory Mendel?
Nah.
I think waktu SPM pun pernah belajar kan? SPM pun? Yeah, so this is more on a flashback ataupun refresh back of your punya understanding lah. Alright, so it's easy. Don't try to memorize, but try to understand. Kalau understand about the concept, it's really much more easier for you. Right? Understand the concept. Don't memorize. Kalau memorize, awak akan pening sikit. Bila ditanya soalan, kalau people twist the questions, it's hard for you to gain the things back lah. Kalau understand or twist macam mana pun soalan, it's easy for you to answer. Right. So some little bit background check about Gregory Mendel lah. Right. His name is Gregory Mendel. The people known him as a father of genetics. Right. He lived in Austria in early 1800s. Interest in math and science, right? Background, grew up in rural areas and works in his family farm before furthering studies, right? Attend University of Vienna, later become a monk in St. Thomas Monastery in Brno, Czech. So dekat sini lah a lot of things happen dekat sini, kat monastery. Alright, so this is where he found out a lot of things regarding the inheritance. Alright, so in monastery he works with peace plants because of his curiosity regarding the factors acting on inheritance. He began conducting scientific studies about the pea plant, and from the study Mendel's law of heredity, dia ada dua benda ni, law of segregation and law of independent assortment. This is from dia punya observation. Right? So I just want you guys to try to bring back, catch back things in mind, bear in mind during this particular time. Masa dia, there's no much peralatan canggih. There's not much understanding on how things works. Macam mana inheritance can be fully understand. Right, so the situation how he go for the curiosity on this particular inheritance is due to the certain things dekat sini. Right, dekat this particular pokok. Mula-mula dia tengok pokok dulu, I mean bunga dulu, and then he goes to peace. When he understand about the bunga, and then he go further with the peace. So let's go back on how it start. Alright, macam mana dia boleh nak tahu macam mana benda ni boleh berlaku and how things can trigger dia punya curiosity lah. Right? Dia punya observation. Right? Dia punya observation first adalah from the particular plant ataupun flowers lah. Right? Traits. Right? Dia nampak traits of the flower, pot color and shape, seed colors and shape, flower color and position and height. Ni adalah traits lah. Ni kat mana yang kita boleh observe. Through breed the plant that have identical characteristics from the parents through each generation. Right. This adalah terminologi lah yang you guys need to understand. Traits ni apa-apa yang that particular organism have. Macam kita cakap pasal kucing tadi. Alright. The fur color, the fur patterns and the breed lah. Right. So if the breed is sama macam dia punya parents, right? Sama macam dia punya parents dia, we can say that there is a true breed. Macam kucing tadi lah, ayah dia color oren, mak dia color oren, memang color oren. Tapi kalau mak dia color oren, bapak dia color oren, tiba-tiba dia color hitam. Right? This must be something wrong. Okay. No, it mean that not something wrong lah. Maybe the traits coming from the ancestor lah, from the grandfather. But for Mendel punya experiments, right? They did a lot of self-fertilization. Right? For contoh ni yang color putih. Self-fertilization means that dekat satu pokok dia tak cross breed ni. Dekat pokok tu juga. So dia breed dekat pokok yang sama, dia nak tengok what is the hasil anak dia. Right? So dia nampak the offspring still color putih juga. And the purple color when they go to the offspring still color purple juga. Right? So there's no changes on the inheritance ataupun heredity of the offspring. So which means that dia punya observation tu, this is the observation. Dia ada set thing in mind, kalau ayah dengan mak dia color putih, anak dia mesti color putih. Ataupun the parents is purple, anak dia mesti color purple. This is dia punya mindset at that time. Right? This is dia punya mindset. So they say is true breed lah. So what happened adalah when he tried to cross breed between the white dengan the purple color. Right? They cross breed, cross fertilization. And it turns out that dia punya first observation tu tak sama. But the first observation dia, with this, the true breed tak sama when you go for the first generation. Right? So, the parents and the purple color and the white colors. And then the first generation, what he found out is that all the flowers turn to purple. Right? So of course lah, if I was him as well, I would think mesti pokok ni akan ada dua color, for example. Alright? So one part is white, one part is purple, for example. But it turned out the white color is lost. The white color is lost. Only the purple color emerge. So this is the first dia punya observation. So come to his surprise, where is the white color goes? Right? Hilang the white color tu. Right, all of them are in the purple colors. Then he tried to self-fertilize this particular generation. Dia nak tengok balik Adakah purple color ini memang purple color juga? Right? So when he self-fertilized of this particular F1 generation, and he found that the majority of the offspring is still purple, but this is the thing which quite amazed me. Dekat mana datang this particular white color again? Right? It skipped the generation and it's emerged again in the next generation. Right? However, this is where we found out the most famous ratio that I think you guys know as well in biology is 3:1 ratio. Right? Most of the offspring are in the purple color and only small parts of the offspring is in the white colors. So most of the ratio is 3:1 ratio. Same goes when we do the experiment as well in our laboratory. Right? Whenever the seeds yang kita nak, majority adalah dia punya true breed lah. We call it wild type. Yang memang adalah very dominant. And low portion is very low dominancy. We call it the recessive. Right? So from here, they skip one generation and it emerged again in this particular generation. And this 3:1 ratio. So what happened adalah, here actually what happened. This is the explanation. The purple thread flower were mainly observed in F1 and F2 generation. And we call it the dominant expression. The purple color. Because the purple will always merge. Right? This is what we call it the dominant punya expression. The white colored threads flower were only observed in the next generation, which is the F2 generation, and it's a very small portion. This is what we call it the recessive expression, right? So the segregation will be 3:1 ratio. This is what I did mention just now, and this is the definition. Dominant is the allele whose genetic expression is masking the expression of the second allele, right? Recessive is the allele whose genetic expression is masked by the dominant allele. Allele is the expression, I mean the traits ataupun the phenotypes, right? So dominant is allele whose genetic expression is masking the expression of the second allele, right? Remember that they mask the allele, and this one, the allele is masked by the dominant allele. So awak hafal satu ni ataupun awak understand one part, we will understand the second part, right? So what happened actually dekat sini, right? What happened actually dekat sini? So R is coming from one parent, capital letter R, and this is a small letter r. So what happened adalah when they crossbreed, one parent give one of their punya traits to the punya offspring. So what happened? This particular F1 generation, they have the capital R and small letter r. So what happened in here is that they have a contain two alleles, one coming from each parent. So however, the R small letter R is being masked by the capital letter R. So capital letter R is the dominant allele. They are masking the small letter R. So that's why all the offspring in the F1 generation in the purple color. So what happened in the F2 generation, right? Most of them are in this particular form, right? The mixture of the alleles, capital letter R and small letter R. So they have these two particular allele. Macam parent dia juga, parent dia ada dua, right? RR dengan R kecil, right? This is as well. So what happened when it cross-fertilized, the self-fertilization of this particular F1 generation together with themselves, right? So this is what happened. R besar dan R kecil is this. R besar dan R kecil is also this one. So what happened when they give capital letter R, one capital letter R, and give one capital letter R, it always turn up to be dominant, like the parents. So what happened this one as well, they give the small letter R, and they give the capital letter R, so it turns out to be big mass, and it also shows the dominancy as well. Same goes with this as well, so it will be shows the dominancy. So what happened adalah when the small letter R provided by the parents and the small letter R as well provided the parents, right? It shows this particular traits, right? So this is the recessive traits that always being masked. So sometimes these particular traits is very berharga lah. You can say it's hard to find, it's hard to get as well. Right, so this is what happened. This is the summary of the observation from the Mendelian theory. So he came out to be that, ini adalah definition allele straight genes. Right, so heterozygous is the an individual with different alleles. Right, this is the heterozygous. Homozygous is an individual with identical allele. So macam parents ni tadi lah. So they have the identical allele. So this is the homozygous. And this is homozygous, and this is homozygous, and this is heterozygous, where they contain two alleles at the same time. Right? So that's why some plant breeder, they love to have this particular pure breed ataupun true breed lah. Dia tak contaminate with any crossing from other parents ataupun from other generation lah. Right? So that's why when they do cross checking, right, they will have this particular homozygous punya traits yang dia nak, yang dia perlukan, right? So they can have them to become dia punya induk lah, alright, untuk dapatkan dia punya hasil. I think same goes with macam kambing ataupun paling senang macam ikan laga lah. We do not want the modern beta, but we want the wild beta atau the true breed beta. Kalau kita dah cross dan kacuk dia dah offspring dia akan berbeza. So you can imply this particular knowledge. Kalau breed ikan beta and so on, this is big, right? So the first law of segregation is diploid organism pass allele for a trait to its offspring is such that the offspring receive one allele from each parent, right? Parents dia akan pass dia punya inheritance by dia akan provide one of the alleles from them to dia punya offspring. Macam ni dia bagi R besar, ni dia bagi R kecil. Dia akan dapat lah. So this is what happened. Alright. So this is dia punya first law of segregation. Right. Diploid organism pass allele for a trait to its offspring in such that offspring receive one allele from each of the parents. Right. Dia akan dapat lah. So this is hasil dia. 3:1 ratio. From this particular experiment, right? From this particular experiment, Mendel tried to assimilate the similar observation in pea plants, right? What actually in the pea plants? Pea plants ni dia macam flower tadi, but dia ada extra features. For example, dia memang dah ada color yang berbeza, tapi dia ada juga rupa paras yang berbeza, right? For example, the pea. Right, dia ada two traits yang berbeza. Seed color dengan shape color of the seeds. Right, satu yellow color with round piece, satu green color with wrinkle piece. So dia nak study lah, adakah sama? It should be the same lah, based on dia punya observation lah. This is what he found. Right, eksperimen yang sama, dia buat yang sama. Kalau piece ni self-fertilize, I mean yellow dengan round, self-fertilize, dia akan keluarkan hasil yang sama juga. Same goes with the green dengan wrinkle. Self-fertilize, dia akan keluar hasil yang sama juga. And then he try to do the crossbreed of the particular offspring. Dia buat crossbreed. Right? So in F1, only yellow and round seed trait observed. So which mean that dia dah tahulah. So which mean that yellow and round is the dominance allele. I think dominance allele lah. So maknanya dia tahu this one, the yellow and the green and wrinkle will be the recessive allele. Right? So will be the recessive. What happened when he do the crossbreed? Right? The crossbreed dia dapat ni. What happened is that they do the self-fertilization of the F1 generation. So of course, if I was him as well, based on my previous observation, I will make a hypothesis. So, okay, it will be 3:1 ratio. Same goes like before. Most of them will have their piece, brown and yellow, and only one wrinkle, for example. Alright. So, this particular hypothesis is very interesting and very important lah. Alright. For example lah, if awak plant breeder, alright, awak ada plant, contohnya awak breeder ikan beta lah. Awak confident lah. Okay, kalau ayah dia color merah, mak dia color merah, anak dia mesti color merah. Tapi tiba-tiba awak dapat 3:1 ratio, tiba-tiba anak yang lagi satu tu, anak ikan tu jadi lagi satu tu, one portion is more on color dia kali hijau. So this must be very very special lah. So of course lah, kalau awak jadi breeder beta pun awak boleh cakap, okay, siapa nak beli beta kali hijau, aku jual harga 2 kali ganda lah, sebab susah nak dapat, for example. So bila awak breed, next time awak dapat inheritance dia kali hijau, awak boleh jual 2 kali ganda. Of course, this is the breed lah yang awak dapat. Next time awak cari buyer, okay, kalau merah ni aku jual harga RM10 for example, kalau hijau aku jual RM20. So next time, okay, tak apa, janji dengan orang nak beli, tak apa, next breed aku jual lagi kat kau, jual RM20. Kau tunggulah. But hipotesis tak apa, memang hipotesis dia ada 3:1 ratio, nanti ada one portion yang akan jadi kali hijau, aku jual. Contoh. If you are plant breeder pun awak akan rasa, okay, the special one, we try to give it to you with this particular price and so on, ataupun you want to have that one lah. Tapi turn out that what happened is different compared to the observation yang dia nampak from before, right? It's 9331, right? The 9 of course dia memang dapat apa yang dia nak lah, right? Dia dapat apa yang dia nak, memang tinggi. The dominancy of the round and wrinkle tu sangat tinggi, round and yellow tu sangat tinggi. It's 9 portion, and the wrinkle and green and wrinkle tu memang 1 portion, very sikit. Tapi come to his surprise, right? There are more new features are emerge, right? Which mean that the mixture of both of these, round and green, wrinkle and yellow, ada keluar. So 3:3 ratio. So this is things are very new. What things are very new from his finding and observation, right? From his finding and observation, right? So when he try to make the Punnett square, I mean the relativity of this particular Punnett square ataupun dia punya dispersion, right? Of Punnett square, he found out that this is the dispersion, right? So of course the capital letter is always be the dominant one, right? So from here you can see like, so based on the first law tadi, allele akan pass by from one parents to another, right, to the offspring, right? So this is the alleles, right? Allele dia adalah R besar, R kecil, Y besar dan Y kecil, alright? So dia akan bagilah salah satu allele ni mesti akan dapat ke anak dia. So contohnya allele R, dia akan bagi salah satu allele R ni. So which one? R besar dengan Y besar dia akan bagi satu. R besar dengan Y kecil pun akan bagi satu. R kecil dengan Y besar pun dia akan bagi satu. R kecil dengan Y kecil pun dia akan bagi satu. So these are the alleles yang akan di pass through kepada anak-anak dia. So when we do the Punnett square of this, right? So this is the dispersion of it, right? Of course R besar ni adalah dia punya shape, right? So this is why the shape color is round, right? Y kecil is the color of the seeds, right? So that's why the color green, right? So this is the new emerge traits, right? R besar, Y kecil. R besar, R kecil, Y kecil. R besar, R kecil, Y kecil. So same goes with this particular yellow and wrinkle. Alright. So this one as well, R kecil, Y kecil is similar with the parents. So from his observation, right? So there's a new traits that emerge, right? New trait that emerge. Then he come up with this particular laws yang baru. When two or more traits are inherited, heredity factors are sought independently during gamete production, meiosis, right? Giving different traits an equal opportunity of occurring together, right? So macam ni lah tadi, alright? Traits ni, they can have randomly assorts, tapi dia akan dia akan pairing lah, by pairing the gametes, right? So this is where we come up with this, alright? So kalau traits dia, kalau is single traits like the flower just now, only the colors, we can directly predict is 3:1 ratio, right? However, if more traits, contohnya macam shape dengan color tadi, small shapes, they will have this particular laws. When two or more traits are inherited, heredity factors are sorted independently during gamete production. Alright, dia akan bercantum lah independently. Dekat mana dia nak RY, R kecil ke, RY kecil besar ke. Right, and then this is giving the traits an equal opportunity untuk keluar. Alright, so this is dia punya experiment. And this is dia punya observation. Of course lah, Mendel dia ada lagi more new theories and so on. But these two particular theories are very, very interesting. Alright, for you guys punya understanding in this particular level dulu lah. Right, so 3-to-1 ratio is more on one single trait, mono traits. But a lot more traits, we call it dihybrid theories lah. A lot of width. And you can see this particular new features of traits. It's quite interesting. Contohnya, okay, breeder beta lagi lah. Kalau dah tahu two traits yang berbeza, satu palakat ke, satu half moon ke, and you can also predict lah half cup dengan palakat dengan half moon dia akan jadi ekor kecil and so on. How things goes lah. Alright. So you can try to use these particular theories on how the inheritance can be. Right, so any questions from here before we go for the last part? Sikit je lagi, then we will end the session. Ada? Tak ada? Alright, give me another 5 minutes and then we end ya. 5 minit lagi kita end ya, Mas. Sorry ya, sikit je. Alright, let's talk about the incomplete dominance dengan codominance. I think you guys understand about this particular term lah, right? So incomplete dominance is the heterozygous condition in which both alleles are partially expressed and often producing an intermediate traits, right? Snapdragon flower, the pink colors, right? R besar, R kecil, R kecil. If we follow the Mendelian theory, of course lah, R besar ni dia akan dominant and shows its colors. But there are certain situations, right? Certain, not all, certain situation, but mostly you know follow that particular pattern lah. But some situation, the incomplete dominance emerge and becoming the pink color here, right? Snapdragon colors lah, the flower punya ni, right? It shows the intermediate of each one of them, right? They not show the red color, but you show the white color as well. So that's why it becoming this. Right? Not all, but you have bear in mind ya, bear in mind dulu lah. The follow the first punya law lah. 3-to-1 ratio, each one of them will shows, and dominant will always masking the recessive one. Right? So incomplete dominance is a condition where the heterozygous condition in which both alleles are fully expressed in the traits. Right? Tadi dia partially expressed. Alright, sikit-sikit so dia jadi intermediate. But this one, both of them is fully expressed lah. Ini dia contoh lah, ni bukan ni betul pun. Alright, so both of them want to be expressed. Right, same goes with the camellia flowers lah. Alright, so the white colors and the red colors of camellia when they crossbreed, ni yang maybe the Mendelian punya hypothesis mungkin, saya tak pasti mungkin. When they cross two different colors, both of them will show lah. Maybe this is the one that he's thinking of, maybe. But this is also the different rare cases where it can shows both of them is showing the both colors, right? The white and the red colors is always being shows, something like that, right? So this is where it shows the codominance of each of the alleles. All right. So last but not least, this is a topic for next week lah. Sikit. Just introduce you guys with the Barbara McClintock, right? Discovery of jumping genes. This call it transposon. So this is very interesting. Just a little bit background of Barbara McClintock, right? Since yesterday or the day before yesterday is the World International Women's Day, right? Happy Women's Day to all. Right, this is Barbara McClintock, studying Cornell University. Interest in cytology in genetics, we call it cytogenetics, similar like Dr. Razik as well, right? The background is in 1940s discovered the transposable element in corns. All right. Latif received the Nobel Prize of Physiology and Medicine. He discovered jumping genes. Genes yang boleh melompat from one side to another side. This is just introduction lah ya. Transposon is segment of DNA that are able to move within the genome, known as jumping genes, move from one genomic location to another genomic location through copy and paste ataupun cut and paste. Dia akan melompat from here to here, from here to here as well. And play a significant role in evolution of many genomes atau mutant strains. Kenapa dia ada dalam Nobel Prize of Physiology and Medicine? Sebab when she discovered this, right? She discovered a lot of disease that related with genetics can be curable. Alright, bila these genes are moving, boleh nampak pattern and traits. Patient are improving, something like that lah. Alright, transposon element is quite interesting adalah because can be used to introduce foreign DNA into a genome. Alright, excuse me. So early on lah, maknanya kita boleh tahu genom ni ada capacity untuk menerima DNA sequence, right? Transgenesis and insertional mutagenesis in different organism. Mana kita boleh letak ada genes masuk ke dalam different type of genome, so we can see the traits apa yang boleh keluar. Alright, so this is useful tools to analyze the regulatory genome, identify genes and the pathway implicated in the disease, pathogenesis or pathogens. Alright, so this is where people use these particular ideas. Alright, dia jumpa these particular ideas, dia jumpa this particular condition, bukan ideas, condition, and people are modifying the finding to become more useful. Macam usahawan lah, bukan businessman, dia macam usahawan. They being creative with this new finding. So people can do a lot of things. People can do genetic modification. People can try to understand maybe bila berubah ke sini asalnya jadi A, tapi bila berubah ke sini jadi B pula. So people try to go and look into that and see what is the differences. Alright, I think that's all for this week. So the 5 minutes is up. Alright, if you guys have any questions like usual, just contact me or communicate with me. I will be there with you guys, inshallah. Alright, so any more questions before we end the session? Any questions, Mas? Any more questions?
No.
No questions regarding the lab, quiz, assignments, quiz tak ada, assignments and presentation. I will let you guys know inshallah as soon as possible later. Right? So wish you guys the best and please stay safe this particular time because a lot of people in surrounds you in this particular phase lah. But don't worry, inshallah everything will be better through times. I think by April kita dah boleh jumpa face to face inshallah. Alright, so let's end the session with tasbih kafarah and salam as. Alright, okay, thank you so much everyone. See you guys again next week inshallah. Have a nice day ahead. Assalamualaikum warahmatullahi wabarakatuh. Thank you all.
Thank you, Doctor.
Thank you, Doctor.
Thank you.
Thank you, Doctor. Thank you, Doctor.
Thank you.